Welcome back to my channel. Every single day, your body is under siege from countless viruses, bacteria, and other microbes. But, thanks to one of evolution's greatest masterpieces, your immune system, you're still here, still healthy. But here's a fundamental mystery. How does this army of defense cells know exactly what to attack and what to protect? Why doesn't your immune system turn on you more often? For years, researchers thought they knew the answer: harmful, self-reactive T cells were eliminated in the thymus, a process called central immune tolerance. But as the 2025 Nobel Prize winners proved, the system is far more complex. Today, we're diving into the revolutionary discovery of the immune system's second, crucial safety net, which earned Mary Brunko, Fred Ramsdell, and Shimon Sakaguchi the Nobel Prize in Physiology or Medicine. They didn't just find a new type of cell, they found the immune system's security guards. 1. The Nobel laureates. Introducing the pioneers. The 2025 Nobel Prize recognizes the groundbreaking work on peripheral immune tolerance. Let's meet the three scientists who laid the foundation for a new field of medicine. Mary Brunko is a senior program manager at the Institute for Systems Biology in Seattle. Fred Ramsdell is a scientific advisor for Sonoma Biotherapeutics in San Francisco. Shimon Sakaguchi is a distinguished professor at the Immunology Frontier Research Center at Osaka University, Japan. Their combined discoveries led to the identification of regulatory T cells, the security guards that prevent immune cells from attacking our own bodies. This knowledge is already fueling new treatments for autoimmune diseases, cancer, and complications from organ transplants. 2. T cells, the body's essential defense force. T cells are central to this story. They are highly specialized white blood cells with critical roles. Helper T cells patrols the body, and when they spot an invading microbe, they alert other immune cells to launch an attack. Killer T cells directly eliminate cells infected by viruses or other pathogens, and they can also attack tumor cells. Every T cell has a unique T cell receptor on its surface. Think of it as a sensor. These receptors are built from random genetic combinations, giving your body the theoretical potential to create over quadrillion different receptors. This guarantees that a cell exists to detect any new threat, like the virus that caused the COVID-19 pandemic. However, this random process inevitably creates some T cells, whose receptors might accidentally bind to your own body's healthy tissue. So, why don't T cells react to your own cells more often? 3. The traditional view. Central tolerance, the thymus test. For years, the established answer was central tolerance. As T cells mature in the thymus, they undergo a test. They are exposed to fragments of the body's own proteins. T cells that react to these self-proteins are flagged and eliminated. T cells that pass the test are allowed to circulate and look for invaders. But what if some self-reactive T cells slip through this initial test? 4. Sakaguchi's insight, the need for suppressor cells. In the 1980s, one researcher dared to challenge the completeness of central tolerance. Shimon Sakaguchi. His inspiration came from a striking experiment. The experiment was researchers surgically removed the thymus from newborn mice. They expected a weaker immune system, but instead, the mice developed severe, uncontrollable autoimmune diseases. Sakaguchi's key observation. Sakaguchi later injected these thymus-less mice with mature T cells from other mice. The injection seemed to protect them from autoimmunity. This suggested a crucial thing within that pool of mature T cells. There had to be a kind of security guard, a cell that could calm and suppress other T cells that had slipped past the thymus and were attacking the body. 5. Defining the regulatory T cell. T cells are identified by proteins on their surface. Helper T cells carry the CD4 protein. Sakaguchi hypothesized that the protective cells he observed were a special subtype of CD4 T cell. After over a decade of work, in 1995, he announced a completely new population of T cells. He showed that the immune calming T cells possess not only the CD4 protein, but also a surface protein called CD25. He named this newly identified population regulatory T cells T. Reg. However, many in the scientific community remain skeptical, waiting for more definitive proof. 6. Brunko and Ramsdell. The scurvy mutation. The definitive proof came from Mary Brunko and Fred Ramsdell, who were working at a biotech company researching autoimmune diseases. They focused on a mutant strain of mice known as scurvy. Scurfy male mice suffered from severe scaling skin, enlarged lymph nodes, and died within weeks due to a rampant, uncontrolled autoimmune reaction. The mutation was known to be on the X chromosome. Finding the exact mutated gene in the vast stretch of DNA was like searching for a single needle in a massive haystack, a daunting task in the 1990s. They eventually narrowed the search down to a region of about 500,000 nucleotides. In 2001, after years of dedication, they identified the defective gene FOXP3. They quickly realized that this mutation was the equivalent of the gene defect found in a rare, severe human autoimmune disorder called IPEX. 7. What is IPEX syndrome? The human cost of a faulty gene. Brunko and Ramsdell's discovery of the FOXP3 gene solved the mystery of this devastating human disease, IPEX syndrome. It's a rare, severe, inherited autoimmune disorder that highlights what happens when the immune system's security guards, the regulatory T cells, fail completely. IPEX stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. The faulty FOXP3 gene means the vital regulatory T cells are absent or dysfunctional. The immune system runs rampant, attacking the body instead of invaders. The primary symptoms are enteropathy, severe, chronic diarrhea and malnutrition, polyendocrinopathy, such as early onset type 1 diabetes, and severe skin issues like eczema. Because the defective gene is on the X chromosome, this severe, often fatal, syndrome almost exclusively affects boys. 8. The final link: regulatory T cells as security guards. The final piece of the puzzle fell into place 2 years later. Sakaguchi and his colleagues demonstrated that the FOXP3 gene is the master key that controls the development and function of the regulatory T cells. A standard T cell that slipped past the thymus central tolerance might mistakenly react to a self-protein. A patrolling regulatory T cell peripheral tolerance intervenes, discovering the attack as a mistake and calming the aggressor T cell down. This prevents autoimmune disease. The discovery showed that regulatory T cells are the body's second safety net, the immune system's security. Detail critical for peripheral immune tolerance outside the thymus. 9. Impact: a new era of medicine. The fundamental knowledge provided by Brunko, Ramsdell, and Sakaguchi is already accelerating the development of new medical treatments. Treating autoimmunity. Researchers are working to induce the production of more regulatory T cells, using substances like interleukin-2, or by isolating and expanding a patient's own Tregs in a lab before injecting them back. The goal is to cure or treat diseases like type 1 diabetes and arthritis. Preventing organ rejection. Regulatory T cells are being tested to prevent the immune system from attacking transplanted organs. Researchers are even engineering Tregs with surface antibodies like a zip code to guide these security guards directly to the transplanted kidney or liver. Fighting cancer. Tumors often recruit large numbers of Tregs to create a protective wall against the immune system's killer T cells. Scientists are now trying to dismantle this wall to allow the immune system to access and destroy the cancer. Mary Brunko, Fred Ramsdell, and Shimon Sakaguchi, through their revolutionary discoveries, have given humanity the fundamental knowledge to control and regulate the immune system. For this, they truly deserve the highest honor. If you found this explanation useful, please hit the like button and subscribe for more science deep dives.